So we're going to show that process. And this chemical reaction, so what we're going to do, what I'm going to do is show the combining of two amino acids, and then you're going to extrapolate that to have several amino acids linked together. And we're going to see that the reaction looks just like amidification because that's what it is. So you have one amino acid, and I'm just going to call this X1. It doesn't matter what it is, it's some amino acid. It could be the same as the next one or not. Okay, and then you take another amino acid. You will have to demonstrate this. So it's not, I don't think it's that hard to do if you just practice. Okay. We'll leave the lone pairs on. Put it there. Okay, so this is going to be amidification. We're going to pop off the water. We're going to pop off the OH off of this amino acid. We're going to pop the H off of one of the H's. It doesn't matter. Okay. This amine is a primary amine, so we end up getting I'm going to draw this down. It doesn't matter. That's the way I usually do it. Okay. So this is called a peptide. That one structure there. So a protein is many peptides all together. But this is the beginning of protein. This bond here is an amide bond, but it's a special kind of bond. Amide bond is called a peptide bond. Okay. So when in the next chapter we'll talk about DNA making protein, and the way it'll work is that there'll be amino acid number 1, and then what'll happen is then you add amino acid number 2, and then over here you add amino acid number 3, and then you go until it's done. Okay, and we'll talk about how it knows when it's done and all that. Okay, so this process continues until the protein, the amino acid is till the protein has all its amino acids linked together. So if you go look back at amidification, you see that it is an amidification. Now you have your primary amine, you have your carboxylic acid, combine them, you form, that's an amide, what we call it, because it just has, we call it a peptide. It's a special name for it. Okay, now, so let's extrapolate this and code it a little bit more. So okay. So this is my amino acid, and so I'm not drawing all the structures. So this is what I just did. Okay. All right. So this is a peptide bond. So what happens is that the protein gets made in that direction. So this would be amino acid number 1. And it keeps on, when we get to talk about DNA, etc., is that the appropriate amino acid gets attached, and it does that until finally you get to your last amino acid. I don't know, we'll call it amino acid 150. Okay. So the amino acid has, so the, right, so the protein's amino acids has a direction and it has a sequence order of amino acids. Now, so this is called the amino acids sequence, and what it is a specific ordering of amino acids from the amine end start to the carboxyl end. So all proteins will start with an amine. So this is a free amine, we call it, because it's over here not linked to anybody. So it's free, it's not attached to anything. And it goes until you get a free carboxyl that's not attached to anything. So the way that you would read this protein is that you read this amino acid first, maybe it's methionine, then an alanine, then phenylalanine, aspartic acid, and so forth. All right. Okay. Okay. Every protein has its own unique amino acid sequence. Okay. And that's so we call this the primary structure of a protein. It codes for the three-dimensional structure. So it itself is in a three-dimensional structure, but it codes. It will tell the structure how to fold up and do its job. Okay. So what happens is primary structure leads to this three-dimensional structure. So the amino acid sequence leads to the actual structure, and the structure leads to its function. So this guy here, if you know this, theoretically you can figure out what a protein does. Okay, that's kind of a cool part about this. All right. Show an example of that. Okay, this represents the amino acid sequence of one of the chains in hemoglobin. So at the top would be this guy over here, valine, and it would have a nitrogen floating around over here. And then what's attached to that is a histidine, and then so forth until you get to this guy here, histidine, and it has a carbon hanging off of it. No other protein has this amino acid sequence. This sequence ends up telling the protein, this guy, to fold up what it does. Now, I told you is that if you just change one of these guys, you can change the whole structure of this. I can't remember which one it is, but it's like this guy right here, maybe this one. You change that amino acids to, I believe it's valine, you get a completely different structure. You get hemoglobin typically kind of looks like that. When you change that guy from a glue to valine, I think it is, it turns into a sickle cell. Okay. So these things are really, really specific. Okay. You just got, and your body knows that, and it's the mistakes, and we got to be careful about that. But by the way, there are safe holds. That's it, because your body does make mistakes. Nature makes mistakes, but it's figured out ways of dealing with those mistakes. Okay. All right. See this in a second. All right. So what happens next, and I'll just show this and then I'll come back to it, is that, so you have your amino acid sequence. It comes off, when it gets built in the ribosome, it comes out as a long chain. Okay, and then what starts to happen is the hydrophobic parts, non-polar amino acids, start to get pushed together. Okay, and what that does is, just like I said before, it pushes, so they go to the core. And then and they start forming London dispersion forces. So inside the core of a protein is mostly London dispersion forces, holding together the abundant non-polar amino acids. Then on the outside will be polar amino acids that are interacting with water through dipole-dipole interactions. And then finally what happens is that some of the polar amino acids get pushed into the middle because the non-polars are just kind of like, and they get pushed in the middle and they pull in some of the polars with them. And that, what happens to them is they link up, okay? And they will form secondary structure, which we talk about hydrogen bonds as well. So let's draw that. So folds up, and most proteins are globular in their shape, kind of spherical. Not all of them, but they usually have some sort of shape that's roundish. That's because of the polar guys. So you have polar amino acids in the middle, and they're held together primarily by London dispersion forces. Okay, just like so. And so it's like the non-polar parts of soap, the tails interact. And then the outside guys, polar amino acids, okay, they interact with water, dipole-dipole. And some polar amino acids get stuck in the middle, and they'll form these things like alpha helixes. And beta strands, which lead to beta sheets. Okay. And these guys are held together by hydrogen bonds, which we want to talk about. Make sure you know what those are. So protein, and this is pretty solid now. So proteins are very stable because of all these interactions. Okay, and they won't fall apart all that easy. They do fall apart, but they not all that easy. Oh, okay. So when a protein folds, we call that the tertiary structure. It's the fold or the three-dimensional structure of the amino acid sequence. It says fold, not five. Still not very good. Okay. All right, let's take a look at these guys here and make sure you know what a hydrogen bond is. So dipole-dipole is a natural interaction between polar molecules. They link up because plus and minus link up with each other. A hydrogen bond is a special kind of dipole-dipole, and it's going to look like this, where one molecule or one part of a molecule is going to have a hydrogen, and that hydrogen is going to have a dipole-dipole. So this can be oxygen or nitrogen. And I'll show you that in a minute. Okay. So this is a dipole. Okay. So that's, you should be able to recognize and tell me what a hydrogen bond is. Really, I love teaching because one thing I love teaching about is that as I teach stuff, I go, oh, I learned stuff. And I just learned something the other day, and it made me go back to my my books to try to figure things out. It turns out, if you look at this hydrogen that's shared, this hydrogen has a covalent bond and a dipole. So it has both kinds of bonds, and that's kind of a cool thing. All right, so what you have here is this hydrogen is being shared between these two guys over here. It's being shared as both a dipole and a covalent bond. So let's go back and look at these things. Okay, so secondary structure. So alpha helices and beta strands sheets are called secondary structure. Okay, let me talk about what we have up here. It says secondary structure is regular recurring arrangement of protein chain. I will ask you on a test, what is secondary structure? I really hate when people write that exactly down as that statement. Because it just looks like to me you just copied it from the PowerPoint. I'd rather you not write it's a regular reoccurring arrangement of protein chain. What it is structure found in a protein that has hydrogen bonds, and it's regular, and it's often found, and it's alpha helix and beta strands. Okay, it just kind of annoys me when people write down exactly the same thing as you find in their notes or in, and they go, well, that's the way you said it. And I go, well, part of it is being to translate things into your own words. And that shows me learning. Is that when you can, I never say the same thing the same way almost. Okay. All right. So let's take a look. Okay, so here, this shows the hydrogen bond. You have this oxygen, this hydrogen is connected to a nitrogen, and then this is a dipole to an oxygen. Okay, and that dipole is a special thing called a hydrogen bond. Alpha helix, okay. Here's a hydrogen bond, NHO, hydrogen bond. Okay, make sure it's, when we talk about these things called beta strands, beta sheets, you need to have a beta, this is the beta sheet. A beta sheet is made out of beta strands. So you have 3 of these making a sheet, just like a bed. A bed sheet is not just a strand, okay? A bed sheet is many strands. A beta sheet is many beta strands, where the strands are held together by hydrogen bonds. All right. Okay, just to give you some, I just like showing you. So here's hemoglobin, okay, it has unique unique fold. The fold is determined by its amino acids. It has mostly alpha helices in the middle. This guy has this thing in the middle, which we'll talk about. It's called a cofactor. This is the biggest protein in the world. It's over 35,000 amino acids long. I said if you had a cup of water, and this was compared to a cup of water, this would be 60 tons. Okay, it's like a giant semi-truck, all right? So proteins can be really, really huge. Go-to guy. Okay, this protein is trypsin. It's involved with breaking down proteins in your intestines. It's just one protein strand, amino acid strand, and you can see that it has the blue is beta strands and the red is alpha helix. Now, all the other coiled stuff is very specific. It's not random. Every time you look at this protein, it will have those green random, looks like coiling. They're not random. That is all structure. Okay, now some proteins, like hemoglobin, require more than one tertiary structure. So hemoglobin has 4 parts to it. It has 2 alphas, or 2 A's and 2 B's. So it has A, B, B. Each one of these is made separately in your cell, and then they come together. Since this protein needs to have more than one tertiary structure, it has quaternary structure. Quaternary structure means you need to have more than one tertiary structure. These tertiary structures can be the same or different. So you have 2 A's and 2 B's. Okay. There's some proteins where, like HIV protease, have the same subunit. So those guys are called subunits. So quaternary structures, the individual tertiary structures are called subunits. HIV protease has one type of subunit and it has 2 of them. They're identical. The protein that I worked on, this protein has 2 types of subunits. It has a green and a red one. Okay, and actually it has 6 greens and 6 reds. You can't see the other set. There's 3 on this side and there's 3 on the other side. And there are 2 here and there are 2 here. So when you add them all up, you have, there's 12 subunits. 2 of each. Then there's 3 of 2 each, 6 of each one.
So each subunit has the same sequence, the same fold?
If they're the same, okay,
It's the same sequence.
like the reds all have the same amino acid sequence. The greens all have the same amino acid sequence. So when the cell makes it, it makes multiple copies and multiple copies come together.
It's the same sequence, it's going to have the same
Folds, right,
bonds.
right, exactly. So each green guy is exactly the same. And then they come together. All right, okay. All right, proteins can unfold, and that's called denaturization. Now, actually, sometimes it's a really big deal. It may be a reason why there's Alzheimer's, just because of unfolding of proteins. Now, sometimes you need to unfold proteins, like when you eat a protein, what you have to do is you have to unravel that protein so that it can be broken up into amino acids. And one way you can do that is by adding acid. I'll go back to the other one. So when the protein gets into your stomach, you have hydrochloric acid. Now hydrochloric acid unfolds it, denatures it, and then a protein comes along, cuts it all up, and sends the amino acids out. Like I said, you could shake up a protein. So if you have milk and you shake it up, you see those bubbles? Those bubbles are denatured proteins. I was doing a hike the other day next to a lake, and you know sometimes you see the little foam that piles up on the side of the shore. That's protein. It's denatured protein. When you pour yourself a beer, you get head on it. The head is denatured protein. Okay, so another way you can do it is heat it. And so if you heat up a protein, it'll unfold as well. And what it does is it doesn't break the protein apart. It just causes it to unravel. Okay, now we get into enzymes. Okay, so enzymes are catalysts. And catalysts are lowered activation energy. Every chemical reaction has to go over a hill of energy before it can happen. What a catalyst does is it doesn't remove the hill completely, but it lowers the hill. And an enzyme is a biological catalyst. Every single chemical reaction in our body needs help, needs an enzyme to help, because they wouldn't have, we just don't have enough energy in our bodies to go over these hills. So what the enzymes do is they lower the hills, and this shows you what happens here. So the red is the normal reaction, and adding the enzyme lowers it. All right, so what I want to do is I want to go through how it does that. Okay. So what you do is you start out with protein and the substrate. So substrate and enzyme. Okay, the enzymes are just here to help. It nothing happens to it during the reaction. I said it's like a midwife. It comes in, helps, and then leaves, and goes help somebody else. So the first step is the binding. And I said that other types of proteins are similar. Like antibodies, they bind. The carrier proteins, they bind. Receptors, they bind. But what enzymes do is they do another thing. They not only bind, but they are active. They actually help out the site. So this is not only a binding site, we call it specifically an active site, because it doesn't only bind things. Like hemoglobin binds something, and then it takes the oxygen, binds the oxygen, takes it to the cell, but it doesn't do anything else. These guys grab a hold of it and then help the reaction take place. Okay. All right, so the substrate is held in the enzyme by London dispersion forces, hydrogen bonds, and or dipole-dipole. So it's docked. So it gets docked in there. And we call this enzyme substrate complex. Okay, once that happens, the enzyme will twist and turn substrate into what we call a transition state. And it's the transition state. Okay, so we have over here our substrate, our product has to go over the hill, okay? This is the activation energy. I said the transition state is sort of in between the two. Okay, so actually the activation energy is what is needed to make the transition state. And then when you make the transition state, it goes flying downhill. So it twists and turns the substrate to turn it into transition state. Okay, and it's locked in there, and that's the enzyme transition state complex. Okay, and then what happens is the activation energy is added, which is lower now, so it's lowered activation energy because they don't need to go as high anymore because you got their enzyme. And then you have your product. So makes the product. Then finally it gets released. And this guy can go back and do the job. All right, so I didn't write it down, but this is the enzyme transition complex. This is the enzyme product complex. And then it releases. And you got
That was a hard drawing.
what's that?
I said that was a hard drawing.
Hmm?
It was a hard drawing.
Um,
I'm running on
Need 2 or 3
the edge
pages.
of my paper, yeah.
Turn the paper sideways. Okay. All right, what I wanted to talk about is some things that affect enzyme activity, okay? Okay, some enzymes need cofactors. And there are 2 types of cofactors. There's coenzymes, and then there's metal ions. Okay, coenzymes come from vitamins. So when you take a vitamin, it gets made into a coenzyme. And so this diagram sort of